Hi everyone. Okay, today we are going to talk about the experiment also. So today I am going to teach you about the experiment. Okay, related to investigation on action of enzyme. Okay, in saliva on starch. So when you go for today's topic, okay, this one will be one of the important topic in our nutrition chapter. Okay, so we know about the nutrition chapter, so we got study about nutrition. So we will be having 7 classes of food. Okay, carbohydrate, protein, fats, vitamins, water, and then we will be having fiber, okay, and also minerals. So when you go for this nutrition chapter, one of the part in the nutrition chapter that we will be studying about is actually on the experiment. Okay, so we got learned in the nutrition chapter that whatever food that we are eating, okay, in that food that we are eating, we will be having fat, carbohydrate and protein, and this fat, carbohydrate and protein will be passing by, okay, our alimentary canal, and it will be digested. So this digestion will be done by the enzymes. So in our body we will be having, okay, some important enzymes which will be working in order to digest the nutrients, and by digesting these nutrients, for sure we will be getting the simplest molecule of the nutrient in order to make it to be absorbed by our blood circulation actually. So whatever food that we are eating will be digested. By doing this digestion, okay, we will be obtaining the simplest, okay, molecule of the nutrient, and these nutrients will be absorbed into our blood circulation and it will be circulated to our body cells. So when you go for one of the enzyme that we are going to investigate today about is the enzyme in our saliva. So when you go for saliva, okay, the saliva that we are producing in the mouth actually contains enzyme, and the name of enzyme, okay, of this enzyme which is produced by the saliva is called as amylase, and sometimes it can be also called as salivary amylase. So when you go for this enzyme, amylase, this enzyme is responsible to digest starch. That means in the food that we are eating, okay, we may have many type of nutrients. Okay, we may have carbohydrate, we may have fats, we may have vitamins, we may have proteins and so on. Okay, so when you go for carbohydrate, okay, carbohydrate is actually can be categorized into many types. So one of the complex form of carbohydrate is actually starch. Starch will be the complex form of carbohydrate, and we are going to learn how the starch is actually being digested by our body. So when you go for complex form of carbohydrate, we got many types of complex form. We are not going to talk about that one today. So we will just see starch. Starch will be, okay, one of the example of complex form of carbohydrate. So this starch is actually also called as complex sugar, and we can find the starch in certain foods such as rice and so on. Okay, so when you go for starch, okay, this starch is being digested, okay, by the salivary amylase, okay, which we can find in our saliva. So our saliva contains amylase. Amylase will be the name of the enzyme. This enzyme going to break down the starch, and when the enzyme breaks down the starch will change into the form of maltose, and this maltose is actually called as simplest or simple sugar. So maltose will be under the category of carbohydrate also, but maltose will be the simplest form of carbohydrate. And when you go for starch, starch will be the complex form of carbohydrate. So amylase is actually doing what? Amylase will be breaking down the starch into the form of maltose. So that is the work done by amylase. And from where we can find out the amylase? We can find the amylase from our saliva. Okay, so this is the thing which is happening in the body. Okay, so we are going to investigate. That means we are going to represent this one in the form of experiment now. So how we can prove that saliva actually contains amylase, and this amylase is actually changing the starch into the form of maltose. This one can be proved by doing this experiment. So if you see the experiment here, this one will be the simplest, okay, apparatus setup that we have to prepare in the lab actually. So when you go for this setup, okay, you can see the red dot stand, and you will be having the tripod stand, and you will be placing the Bunsen burner here, and we are going to fill up a beaker full of water. Okay, and we are going to use two test tubes, and we are going to label the test tube, okay, as test tube P and test tube Q, because the contents of the test tube will be different here. So in the first test tube, you are going to fill up 10 milliliter of starch and also 4 milliliter of saliva. So this saliva will be coming, okay, from yourself. So if you other one is the one, if we are preparing the apparatus setup here, means we are going to do what? We are going to use our saliva. Okay, so we have to take about 4 milliliter of our saliva. We just have to rinse our mouth using water, and we are going to use, okay, that saliva, okay, about 4 milliliter, okay, and we are going to mix it together with the starch actually. Okay, and then we are going to prepare another test tube, and we are going to label this test tube as Q. In this test tube, you are just going to put 10 milliliter of starch and also 4 milliliter of distilled water. We must know that distilled water does not contain anything, but in saliva we will be having the enzyme which is called as amylase. So in the first test tube P, we will be having starch together with the enzyme, okay, which is called as amylase, because you already added saliva. In the second test tube Q, you will be having starch together with the distilled water, and in this distilled water you won't be having any enzyme. So both of the P and Q test tube will be placed in the beaker, and the beaker will be heated by using Bunsen burner. So this kind of burning process, heating process is called as water bath. So we are going to make sure that the water inside the beaker reaches 37 degrees Celsius. Okay, so the water bath must be under 37 degrees Celsius. And why the water bath must be under 37 degrees Celsius? Because we must know one thing: saliva, okay, will be coming from the human body, and the saliva will be under the temperature of 37 degrees Celsius, which is equals to the normal body temperature. So the enzyme in the saliva will be working the best under 37 degrees Celsius. So we are going to prepare a temperature which equals to 37 degrees Celsius, which will be the optimum temperature for the amylase to work on. So we are going to make sure that the water bath is actually under 37 degrees Celsius. Okay, after that we will do what? After 1 hour or 2 hours, okay, or 1 hour or half an hour also will be enough. So after that we are going to take the sample, okay, of the mixture from P and the sample from Q, okay, and we are going to test it. So the test that you are going to take here is called as iodine test and also Benedict test. So when you go for iodine test here, okay, before I talk about the ways how to take the test, okay, I will just introduce what is iodine test and Benedict's test actually. So when you go for iodine test here, iodine test is actually done for the presence of starch. Okay, normally we will be taking this iodine test in order to see whether any food or any liquids or anything contains starch or not. Other than that, Benedict's test, this one is actually done for the presence of simple sugar. If any food contains simple sugar such as maltose, okay, the Benedict test will be giving you a positive result. So if you see here, for iodine test, let's say the food contains starch, means, okay, the original color of iodine will be yellowish brown. Yellowish brown will be the original color of iodine. So if the food or any substances that we are using for the test contains starch, means what will happen? The yellowish brown color of iodine will be changing into blue-black solution. So the color will changes to blue-black, which will be showing the positive result for the presence of starch. Okay, in this case, Benedict's test, okay, will be for simple sugar. So if any substances that we are going to use for the test, okay, contain simple sugar means, okay, the original color of Benedict's solution will be blue, and this blue color will be changing into brick red precipitate. So, if the blue solution of Benedict changes to brick red precipitate means it shows positive result for the presence of simple sugar. So, in this case, okay, after the experiment, okay, I will take, okay, after the experiment I will be taking the sample from P and sample from Q, and I will be doing the test actually. So when you go for experiment P, when you go for solution P, okay, we will take the solution, okay, from P, and we will be doing iodine test. Okay, when you do iodine test from solution P, before the experiment you will be having, okay, you will be having two results here. One is before the experiment, another one will be after the experiment. When you go for before the experiment, we will take the solution P and we will be doing two tests. Okay, the first test will be iodine test, the second test will be Benedict's test. So when you go for iodine test here, okay, this one will be for P here. Okay, for solution P, when you are taking the iodine test for solution P, before the experiment, for sure what will happen, the iodine test will be giving you positive result. Okay, it will be giving positive result. That means, okay, you will be getting blue black solution there. Okay, why is it so? Because you have starch, you got saliva, but before you start the experiment, the starch will be not broken up into the form of maltose by the amylase. So, before the experiment, for sure there will be starch present. So, because of the presence of starch, iodine will be changing into the form of blue-black solution when the solution P is tested with iodine solution. So, that is the reason why it's giving you positive result here. Okay, when you go for after the experiment, when you take the same iodine test here, okay, I am taking the same iodine test after the experiment, maybe half an hour or 1 hour after the experiment, I will take the I will take sample from the solution P, and when I use the sample from solution P and I test it using iodine, okay, I won't be getting any positive result, I will get only negative result. That means the iodine test, I will be using iodine solution to take the iodine test, okay. So the color remains the same, okay, the color will be in yellowish brown. The original color of iodine will be yellowish brown. So the iodine test will be also showing the yellowish brown color only. That means the color does not undergo any changes. Okay, this is showing that there is no starch present here at all. Why is it so? Because the starch together with the saliva, which is being mixed up with the enzyme, okay, called as amylase, okay, will be broken up. That means the starch will be broken into maltose. So you won't be having any starch molecules here at the end of the experiment. Okay, so when you go for the same solution P, okay, and now we are going to take Benedict test also. When you go for Benedict's test at the beginning of the experiment, okay, before the experiment starts, at that time Benedict test will be giving you negative result. That means the original color of Benedict solution will be blue. So after taking the test, the color remains blue. Okay, there is no changes at all, because you won't be having any simple sugar here, because you will be having only starch before the experiment. Okay, but right after the experiment, what will happen? when you take the sample after 1 hour or 2 hours of the experiment, when you take the sample and you test it, okay, and you testing it with Benedict solution, at that time what will happen, you will be getting positive result for Benedict test. Okay, that means the blue solution of Benedict will be changing into brick red precipitate. So in this case, brick red precipitate will be formed. This one is showing that the starch has been changed into the form of maltose. So brick red precipitate will be formed. Okay, so this one will be the result of the experiment, okay, when we tested using iodine and also Benedict. Okay, in this case, if we take the same test for Q, in Q we will be having starch and also distilled water only. So in Q, the result will be very simple. Before the experiment and after the experiment, if I take the sample from Q and I test it with iodine, before and after, the result for iodine test will be still positive. That means the original color of iodine will be yellowish brown. It will be changing into blue black solution. So it will be giving positive result and positive result before and after. Why is it so? Because before and after, the solution will still contain starch and the starch will be not broken up into maltose at all, because you mix it together with distilled water. In distilled water you won't have any enzyme. Okay, so that's why iodine solution will be giving a positive result for the presence of starch. But if you take sample of Q and you go for Benedict's test now, when you go for Benedict's test, okay, what will happen? The Benedict test will be giving us only negative result, because there is no digestion taking place here. The starch is not broken up into maltose at all. So once the starch is not breaking into maltose, you won't be getting any positive result for Benedict test. So this one will be the overall investigation to investigate, okay, the action of enzyme in saliva on starch. Thank you for watching.